good to join you again in the second part of our conference. I know that a lot of you are really eager for this session to attend and learn a lot more about what we already know. COVID-19 vaccine has been an issue of discussion for doctors and non-physicians and the whole public. And it gives me a great pleasure to have Professor Brian Ward with us today. Professor Brian Ward is from McGill University in Montreal, Canada. He was trained at the McGill, the University of London, and Johns Hopkins. He is Professor of Infectious Disease and Tropical Medicines, and he's currently chairs of the Scientific Advisory Board for the Institute of Infection and Immunity of the Canadian Institute of Health and Health Research, the CIHR. He has published more than 270 peer-reviewed manuscripts thus far. A few months ago, Professor Ward gave us a very nice talk, 10 messages on COVID-19 vaccines. And here we go 4 or 5 months later, listening to Professor Ward and what he has to say. Are you going to change your messages, Professor? Are the messages still the same? Do you have new evidence for us to share with the audience? Professor Ward, the floor is yours.
Thank you very much for the invitation. Some of the messages have changed and some have remained the same. This field is changing so quickly that I actually took the opportunity to change some of the slides just this morning from information that arrived yesterday. So I'll walk you through. I should also say that in addition to my work at McGill, I'm on loan to Medicago Inc., which is a small Canadian company that is developing a COVID vaccine that's entering into phase 3 clinical trials in 2 weeks. So it's been a very busy time. As I said, some of the messages have changed, some are the same. The first, and this is a series of questions and answers really. The first question is how many doses of vaccine are we going to need? And the answer is pretty straightforward. It's now very clear, particularly with the arrival of the new variants, that 2 doses are going to be necessary for most vaccines. Since there are 7.8 billion people on the planet and we need to vaccinate everybody, we're going to need 15.6 billion doses. So even though 1 billion doses from this manufacturer or that manufacturer sounds like a lot, in fact, this isn't a situation of winners and losers or one vaccine that's going to take over. We need many of the vaccines to work and work quickly. Most of the estimates of 1 billion or 2 billion doses of vaccine are in a certain time frame within 12 to 18 months. And so if we actually want to get control of this pandemic quickly, we will need many of these vaccines to work and at large scale. Question number 2, are we going to be okay? And will this be over soon? Are we going to die? Well, no, we're not going to die. However, there are still enormous questions that remain unanswered. In particular, many of these vaccines being rolled out are quite different one from the other. And there's at least a theoretical risk of vaccine-enhanced disease. This has been demonstrated with SARS-1 and with MERS. And there are several different ways that a vaccine could result in more serious disease upon exposure to the wild-type virus. And right now, in some environments, exposure to the wild-type virus is extremely high. 50, 60, 70% in some places in South Africa and Brazil, for example. This could happen by Th2 deviation, by facilitated viral entry. These things have happened with other vaccines, and they happen as the immunity from the vaccine wanes. At the current time, for the most part, we only have a couple of months information for these vaccines. So it's entirely possible, even though we haven't seen vaccine-enhanced disease yet, that we will see this as the immunity from the vaccines wanes. And as you know, the vaccine development process for COVID has been dramatically compressed from the normal 10 to 15 years down to under 18 months. And of course, that means that there are increased risks, even if you do everything very carefully. So the second message really is that vaccine development is remarkably compressed. Problems will happen. We've already seen anaphylaxis with one of the 2 mRNA vaccines, which is about an order of magnitude higher than normal. So normally we see about 1 per million anaphylaxis cases with all vaccines. And with the Pfizer vaccine, we've already seen 11 per million. There have been reports of neurologic complications. There's no evidence of vaccine-enhanced disease so far, but we have to actually wait and see. This possibility of vaccine-enhanced disease is thought to be worst for for vaccines that are inactivated whole virion plus alum. So some of the Chinese vaccines may give us a first look at whether or not there might be vaccine-enhanced disease. But the most important thing is that we need to figure out how to communicate these risks as we roll forward and we have waning immunity from vaccines. Question number 3, what's here and what's coming? Well, everyone is making a vaccine. Major pharma, small companies that want to grow, academics, and even your grandmother. Many people have vaccine trackers, and at the current time, this was pulled off one of the vaccine trackers just a few days ago. 64 that have actually arrived in clinical trial development, and a number of others, another 100 or so, that are in preclinical development. The front runners you see down below that are in clinical development, and I'm very happy to say that the company that I work with is the only virus-like particle that is actually on this list. And entering into its phase 3 study in the next couple of weeks. These are the estimated doses that people say they can produce by the end of 2021, but I'm sure you're all aware that this just isn't going to be the case. Already, several of the manufacturers have run into enormous problems supplying the doses of vaccine that they said they could. Right now, Canada has not received any of the vaccines it was promised for the last 2 weeks. This is, of course, an enormous problem for the rollout of a 2-dose vaccine strategy. And even at 8 to 9 billion doses by the end of 21, I've already said that we're going to need twice as many doses if we want to vaccinate everybody in the world. So clearly the numbers become very important. Furthermore, some big names are dropping out already. This is just a partial list of the people who are most advanced. And you can see that one of the early front runners, the Queensland vaccine, has already gone because their vaccine made people seroconvert to HIV. 2 weeks ago, Sanofi announced that it was getting out of the race, and it's actually given over its production facilities to Pfizer to produce their vaccines. And just last week, Merck announced that neither of its 2 live chimeric vaccines was going to move forward. These are not the last of the vaccines that are going to implode on their way towards clinical licensure. So message number 3, the situation is already complex, and it's going to get more complex as different products are licensed in different places. And furthermore, these products will not be the same in terms of their ease of use. We've already seen that. Refrigerator versus -70 freezer, those are huge differences. Side effect profiles are already starting to be quite different, as you see with the mRNA vaccines and anaphylaxis. Short-term efficacy is also different, and I'll show you a slide from the COVAX presentation yesterday, and durability of protection is completely unknown. And of course, the wild card of the variants is coming up. So which vaccine is best? Well, we're clearly talking about not all apples or all cherries. We're talking about apples, oranges, and bananas for some of these vaccines. So far, the primate and clinical data for many of these candidates shows really good protection. Early after vaccination, range 50 to 95%. And very reassuringly, all of these vaccines show their best protection against the worst outcomes, which is very good. However, for several of the vaccine candidates, the protection of the deep tissues is better than the protection against in the respiratory tissue. So for example, the adenovirus vaccines appear to be able to protect the lungs, but in the upper airways, you still have virus growing. And so how well will they protect against transmission? And what is called sterile immunity, i.e. the complete protection against any infection, is actually very rare and unlikely to occur. Nonetheless, these kinds of data, this is from the Pfizer trial, are really promising, at least against the original strains. Okay, so message number 4 is nobody knows. We're not sure what a good vaccine should do, and immune correlates will not be the same for the different vaccines. We will have first, second, and third wave vaccines. And in fact, we need them. And clearly, many of us are chasing neutralizing antibodies, and I'll show you a slide in a minute. But it's important to recognize that neutralizing antibodies are highest in those who do clinically very poorly. And many asymptomatic cases make little to no antibody response. So clearly, neutralizing antibodies are not everything. So what kind of immune response is good? And is there a correlate of protection? Well, you need all of the above, not just antibodies or cellular immunity or innate immunity. There's good evidence that the absence of type 1 interferons lead to bad outcomes. This is from Casanova's group, recently published in Science. If you have problems with your type 1 interferon, you actually end up in the ICU or in hospital. On the other hand, too much cytokine can lead to cytokine storm. High titers of neutralizing antibodies are good, we believe. Convalescent plasma works. And Dan Baruch has just published some lovely data in primates showing that high titers of antibodies can actually prevent a viral infection of both the upper and lower airways. But clearly, cellular responses are important. Particularly, we know this from asymptomatic subjects and animal models. So the answer is yes, probably all of the above, but we really don't know yet what is the most important. And these are data that were presented yesterday at the COVAX webinar by Dr. Dull. And there was a lot of talk about correlates of protection. And these are data that Dr. Dull has compiled from published and unpublished data looking at the different vaccines versus convalescent serum. So on the right-hand side of each of these graphs for these different vaccines, you can see the convalescent sera and how well the different vaccines have done. And there is actually a pretty good correlation between the fold increase, the comparability of the neutralizing antibodies induced by the vaccines and their efficacy. So you see, as you go from 6-fold lower in the Sinovac vaccine all the way up to 3.8-fold higher in the Pfizer vaccine, you see a steady increase in the efficacy of these vaccines. And from our point of view, very reassuringly, the Medicago data from the phase 1 study shows that our vaccine elicits antibodies that are 14-fold higher than a panel of convalescent sera. So we're actually quite optimistic. But this is the beginning of an evaluation of a correlate of protection. So question number 6, will vaccines be distributed equitably? And this is a facetious map of world happiness. And you can see that, in fact, you guys in the Middle East can be actually quite happy for a variety of reasons, as we are in Canada. However, availability of vaccines will make a big difference to how happy people remain. And so the simple answer to the question, will they be distributed equitably, is no. Already, we see huge differences between the vaccines that are available and being distributed in countries like Canada, the US, Europe, United Arab Emirates, and others, while large areas of the world are simply not serviced. They're not getting the vaccines. And for example, Moderna, one of the front runners, just announced last week that it would not be supplying vaccines to COVAX or to Africa, full stop. So Moderna opted out just a couple of days ago. And of the more than 12 million doses of the COVID vaccines that were distributed in early January to 33 countries, none had been distributed to Africa. Okay, so this is actually a huge big deal because we have to solve this problem globally. Another related question is, should companies profit from the distribution of these vaccines? And some large companies like AstraZeneca have said, well, we'll sell these at cost. We'll only charge $5. But if you're making 2 billion doses of vaccine, multiply that by 5, and you still have a very large amount of money. And there are large differences between the small companies like Moderna that has no cash flow and the larger companies like Pfizer and AstraZeneca that have millions of dollars per day in profits. So small companies cannot offer their vaccines at cost, while larger companies can actually take a more liberal view of costing their vaccines. No matter what happens, SARS is not going to respect borders. It already hasn't. And failure to vaccinate the poorest will guarantee the continued presence of the virus and increase the risk of variants, which we're already seeing. We have to balance the pressure to vaccinate at home and profit considerations against global public health. And the size of the companies will influence these decisions. Okay, Question number 7, how best to use the doses available? And it's really interesting to see people changing almost from week to week. And we have to recognize the quote from David Heymann at the WHO has never been more true than it is now, that with vaccines, we're not only building the boat as we're sailing it, but we're actually learning how to sail because things change so quickly. So who should be prioritized? Can lower doses be used? What about only one dose? Is the interval fixed between doses? And can different vaccines be used? And these are my opinions. But prioritization clearly varies by region, and queue jumping behavior is already happening. Just last week, we had a report in Canada of a very wealthy country chartering an airplane to go up to the Arctic because we've prioritized the indigenous communities in the north. So this rich couple flew up to the Arctic to get their vaccines. Can lower doses be used? Well, Oxford AstraZeneca's mistake in using a lower dose in their clinical trial suggested that some of the vaccines can certainly be used at lower doses, but we really have to go with evidence-based medicine in most cases. And that's why the United Kingdom did not authorize the half-dose, full-dose regimen that was the Oxford AstraZeneca mistake. What about only one dose? Well, the data is surprisingly good with the mRNA vaccines at least, but clearly with the new variants, we now know that higher titers of neutralizing antibodies are really important. And so one dose for most of these vaccines is probably not going to be good enough. Is the interval fixed? Well, yeah, probably not. I mean, right now, that's how they're being tested. But in fact, there's really no data for whether we can prolong the interval, but at least up to 2 months interval is probably okay, and maybe as much as 3 months. However, the immune response is likely to be different if we do that. And so we're moving into the area of conjecture as opposed to evidence-based medicine. Can different vaccines be used? Again, probably yes. But currently, the only data for heterologous prime boost are mistakes. There's very good immunologic data suggesting from animals that this might actually be the best strategy if both of the vaccines target the same antigen. But there's a large study that's only just starting in the UK right now that's testing adenovirus followed by RNA virus or RNA virus followed by adenovirus. So we will find out in the next couple of months whether this idea of heterologous prime boost has some validity. Okay, the wild card that has been thrown into this whole thing is the emergence of variant strains. More than 317 mutations have been defined, and some of them are particularly dangerous looking in terms of transmission and also in terms of resistance. For example, the South African variants they can completely evade neutralization by some of the most potent monoclonal antibodies that have been tested. This has implications for diagnosis, for therapies with monoclonal antibodies, and also for the vaccines. And these are data also from next strain, and you can see that already there is enormous heterogeneity of the strains around the world. And I advise you to go and look at this because it's really quite sobering. Geographic distribution of variants is already widespread, and we actually have increasing viral phylogeny differences between these different regions. And you can see that the South African, Brazilian, and UK strains are already very common. They have taken over transmission in some of the countries where they have emerged. This will have implications. These are data presented yesterday by Dr. Maddy and are on the Novavax website. Novavax was conducting its phase 3 trial in South Africa. And you can see that the orange is the arrival of the South African variant. And you can see that a large majority of the cases in their study were in fact this new variant that is quite resistant to some of the monoclonal antibodies. And their overall vaccine efficacy was about 89%, according to the press release, but their efficacy against the new variants was under 50%. So these will have impact on the evaluation of the coming vaccines. So the answer, global scale experimentation is already underway. There isn't a single formula for success, Government and public health authorities are under enormous pressure to get it right, but they don't know what's coming. So it's very easy to criticize during this process of changing advice. But at the current time, evidence-based decisions, even the best guesses, are being made in good faith and should be respected. So for example, the UK and one Canadian province decided that a larger interval between first and second dose might be appropriate to allow more people to receive their first dose. But in light of these new emerging and more resistant and transmissible variants, that advice may no longer be good. And those decisions were only made a couple of weeks ago. Question number 8, should vaccines be used as political tools? And the answer is they already are. Vaccines have always been predicted to be political, and that's exactly what's happening now. This is actually particularly true in the maldistribution of vaccines around the world. And the WHO is actually really angry with Western drug makers because many of them are distributing their vaccines almost exclusively to wealthier countries. And as you can see here, Moderna is 100% going to wealthy countries. Pfizer is mostly going to wealthy countries. Oxford AstraZeneca is distributing its vaccine more widely with a fairly large proportion going to COVAX. But in fact, it's mostly the Chinese vaccines that are filling the void. Message number 8, for whatever reasons, China is filling the void for the moment. COVAX eventually may help to address this, but this is entirely dependent upon the goodwill of wealthier nations, which is a very weak position for COVAX to be in. Question and message number 9. Initially, the anti-vaccine networks were very quiet, but they have already had enormous influence, and this is getting worse as adverse events start to be reported. Currently, we have about 30 to 40% hesitancy in some places, and as more reports of anaphylaxis or neurologic consequences, even though they're rare, this will get worse. Question number 10, how long will protection last and are next generation covaccines needed? And the answer is clearly yes. This is the emergence of the so-called B.1.17 mutation, a highly transmissible variant in the UK. Oliver Morgan presented this a couple of days ago in a WHO consultation. And you can see that this variant went from 0 isolations to representing more than 99.9% of the isolates in the UK in a period of under 5 weeks. So bottom line, RNA viruses mutate. They mutate faster when under immune pressure. And in some areas, the immune pressure is getting to be very high. More than 300 mutations have already been described. The nomenclature is really a mess. And people are trying to get away from calling things by geographic names, UK, Brazilian, and so on, but there isn't an agreement yet. Clearly, there are some mutations that are really bad, like the E484K mutation that causes a 90% reduction in neutralization. And as I said, there are huge implications for diagnosis and transmission and vaccine efficacy. So priority targets in most places in the world, the elderly, those with comorbidities, and healthy adults. We have no data for neonates, kids under 5, and pregnant women yet. And so most places are vaccinating in this kind of order of priority. But in fact, these areas, these groups that are not prioritized may play a disproportionate role in transmission. And vaccines are clearly subject to moving goalposts at the current time. The first phase vaccines in phase 3 trials took 3 to 4 months to show efficacy. Because of the higher transmission rates, the Novavax study was able to actually reach its interim analysis in under a month and a half. Second wave vaccines, however, will need to move into a very complicated environment with new variants, with vaccines already available, and the pool of elderly and individuals with comorbid conditions rapidly shrinking. In the absence of a correlative protection, we need to do placebo-controlled trials, but now there are ethical constraints that were not there before. And so many companies, including ours, are moving to a blind crossover trial design. So in conclusion, the way we see the world is massively changing, and it is changing literally from day to day. Thank you for your attention, and I'd be happy to take any questions.
Thank you very much, Professor. Yes, we have lots of questions. Are you ready for this?
Ready for the onslaught.
Okay, Professor, I'll start with some of the common questions that have come across the talk and during the day. I took my second dose of the vaccine, and it just happens that I did not develop neutralizing antibodies. shall I take a third dose from a different vaccine or a different company?
Well, so my short answer to that is absolutely yes. That is exactly what I would do. And I think that there was great discussion yesterday about the potential benefits and the small risks of what's called heterologous prime boost. There's very good immunologic evidence from a number of other diseases that heterologous prime boost may in fact be a very good strategy. And that's why the UK, the Oxford group, is just starting to study that. And they will be adding other vaccines to their study of using an RNA vaccine first, a DNA vaccine second. And when a protein adjuvant vaccine like ours is licensed, the Oxford group will add our vaccine, I hope.
How long would you wait if I have antibodies and I see them declining? Shall I wait until they are negative to take a boost? People are talking about annual vaccine like flu vaccine. What's your take on that?
We don't know. We have no idea. There's, as I said, the level of antibodies in people who are asymptomatic, in many cases, is close to zero in terms of neutralizing antibodies. And you may be one of those people. You may be a person who develops mostly a cellular immune response. And those cellular immune responses are actually really effective, and they're quite durable. However, at the current time, we don't have a cellular correlative immunity, and they're complicated to measure. So far, neutralizing antibodies appear to be the best or the most likely correlative protection, but we have no idea how long they last or whether there will be an anamnestic response to a booster dose, whether with the same vaccine or with a different vaccine.
So there is not a big red flag for somebody, for example, to go from a Sinopharm vaccine and taking a Pfizer vaccine after some time?
Well, the obvious answer to that question is the obvious answer to that question is that you put yourself at risk for the adverse events that are particular to each of those vaccines, and clearly they will be additive, right? So that if each vaccine has its own spectrum of adverse events, and so if your plan is to take two different vaccines, you are then subjecting yourself to the cumulative adverse events from those two different formulations.
If I get COVID infection and my infection was mild, there are experts encouraging people, even if they're just recovered from COVID infection, to take the vaccine. How soon would you think a reasonable time is to wait before you take the COVID vaccine after COVID infection? Some people say 3 months you have to wait. Some people say you have to check the titer of the antibody. And some people say, if you haven't been to the ICU, you probably don't have a high titer, just take the vaccine.
Well, that's certainly the approach that's being taken in the clinical trials right now, in that people are vaccinating whether people are seropositive for COVID or not. And so far, it appears that individuals who have had COVID disease, mildly symptomatic or asymptomatic, do boost their immune response to the vaccines. I don't think it really matters. And I would say probably at least a month. But right now, these large studies that are moving into areas where there's been a fairly high rate of transmission, like Brazil and South Africa, people are stratifying their outcomes based on their serostatus at the time of immunization, and you're getting a good response. So I think that the vaccines will do something different in some people from what the natural disease does.
Sure. I took the first shot, and after the first shot, I contracted the COVID vaccine. Shall I continue with the second shot?
Probably with the same caveat of waiting a little bit of time. The immune system doesn't work from day to day. The immune system typically takes a month or two to make a fully mature response, sometimes even longer. And so you probably get your best bang for the injection, the best immunologic bang, if you wait a month or two.
So this is somebody who took the first shot of vaccine, got infected.
Yep.
then wait for a month or two, and then take the second shot?
Well, yeah, you're asking questions that nobody knows the real answer to. So what I'm saying is my personal opinion.
Fair enough.
But it is, there's pretty good evidence, for example, that the shingles vaccine does something different from natural disease. So even older people who've had shingles are advised to get the vaccine. And I think we will be in a similar situation with COVID-19, that the vaccines will provide protection, some of them anyways, in a different way from the natural disease.
Well, I totally agree with you. And excuse me if I was asking questions that I know science did not answer yet. But the reason for me to ask these questions, Professor, was basically try and reassure myself, my colleagues, and just make sure that no big contraindication is being crossed here.
Well, I mean, obviously, each of these vaccines comes with side effects. You've been vaccinated, so you probably know that your arm hurt, and you may have even had a mild flu syndrome. It is not completely benign to take the vaccines, and so we should not be using them indiscriminately.
And last but not least, what's your take on the vaccine-enhanced disease? A lot of us got vaccinated, and we definitely are seeing our colleagues who got vaccinated infected. Is this because, do you think they're reaching this stage of vaccine-enhanced disease? How soon after the vaccine do you think this is developing as an entity?
Well, so first of all, it's important to say that the vaccine-enhanced disease right now remains completely theoretical. It's based on animal models from SARS-1 and MERS. So, and there are biological differences between SARS-2 and SARS-1. SARS-1 infected the macrophages quite well, and macrophages have been implicated in vaccine-enhanced disease. SARS-2 does not appear to productively infect the macrophages. So this may be a worry that never comes to fruition, but we need to monitor this. And there's the CEPI-Brighton collaboration task force on vaccine-enhanced disease, on which I sit, has just had its case definition for vaccine-enhanced disease accepted by vaccine. And so that will be released in the next short while.
Professor, what time is it there in Montreal?
It is 25 to 6 in the morning.
6 in the morning. My dear colleagues, Professor Ward, waking up for us before 5 o'clock in the morning to share his thoughts and knowledge with us. Professor, we're very appreciative for your commitment and your sharing with us your experiences. It's been an honor to have you, sir.
It's been a pleasure. Thank you for the invitation.
Thank you very much.
Good luck with the rest of the conference.
Thanks a lot. Thank you.